Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 5 from our Campbell's Biology 12th edition. So let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
Wiki? Oh, Wiki! Get... Oh gosh, guys. Wiki! You're gonna knock everything over, Wiki. All right, with that crisis averted, let's go ahead with Chapter 5. This chapter deals with the structure and function of the large biomolecules. These biomolecules include proteins, they include carbohydrates, they include nucleic acids, the DNA and the RNA, and they include lipids. So when I say biomolecules, these are the four classes of biomolecules. Biomolecules include carbohydrates, lipids, proteins, and the nucleic acids. And three of these four are known as macromolecules for their ability to form polymers. The three biomolecules which qualify as macromolecules are carbohydrates, proteins, and nucleic acids. So if I were to ask you which of the biomolecules is not classed as a macromolecule, what would you say? That's right. The lipids are not macromolecules because they do not form large polymers. The rest of these are made up of monomers, that means building blocks, if you will, that link together covalently to form polymers. Okay, so again, monomers, the macromolecules have monomers, which are the repeating units that serve as building blocks, and the macromolecules can use those monomers to link them together to form polymers, which are long molecules consisting of many building blocks. Oh, Wiki's here scratching the monitor. Hi, Wiki. Hi, Wicket. Oh gosh, I'm sure this is not working with everybody. The microphone. I love you too, buddy. Gosh, Wicket really wants to make a cameo today, you guys. So let me just maybe put him on the couch over there so he's not getting in everything. Hi, Wicket. Oh my gosh. Okay. Oh, poor Wiki. He just wants attention today. Oh gosh, I don't know if this is gonna be in a clip at the end or something, like one of those cutscenes. Oh gosh. You're so fun. Wiki. Wiki. Oh gosh. Wiki. Yes, say hi to your adoring fans. He just puts his tail in my face. Wiki. Alright, you go sit on the couch. Okay, bud. Alright, sorry, you guys. My microphone's way over here now. Sorry about that. Alright, let's carry on. So these monomers that I told you about with the macromolecules, these monomers are building blocks which link together to form polymers. And the process by which monomers link together is known as dehydration reaction, sometimes called dehydration synthesis, or even condensation reaction. This is the process by which the monomers for the macromolecules link together to form covalent bonds so that they can form polymers, okay? And the converse of that, the opposite of that, is hydrolysis. Hydrolysis is how these polymers are broken back down into monomers by breaking those covalent bonds between the monomers. So let me show you what I'm talking about. Look at this. at the top here, we have an example of dehydration reaction. And right below it, we have an example of hydrolysis. So let's start with dehydration reaction. Take a look here. Here you have a short polymer. So it looks like you have 1, 2, 3 monomers that have already been linked together. And you want to link on a fourth monomer. Here's the unlinked monomer. So how do we link this monomer to the growing polymer chain? You want to form a covalent bond between them. And the way you do that is by removing a hydroxyl group from one of these entities and a hydrogen from the other entity. So what happens when you remove two H's and an O? You remove two H's and an O, and that means you have removed water. And the result is a covalent bond forms between the entities. And now you have 1, 2, 3, 4 monomers all linked together as a polymer. That's a longer polymer. And if you want to extend this polymer even more, you can do that. You just keep doing the same process over and over again. And do you see how you had to remove a molecule of water in order to synthesize that bond? This is why it's called dehydration reaction, because you're removing water, sometimes called dehydration synthesis, because you're synthesizing a new covalent bond. And it does not matter if you're talking about dehydration reaction in proteins. You could link amino acids together to make proteins. For nucleic acids, you link nucleotides together to form DNA or RNA. And for carbohydrates, you link monosaccharides together to form polysaccharides. These are all linked together the same way. They're all linked together with dehydration reactions. So it doesn't matter which monomers you're talking about. All of the macromolecules have building blocks, and those building blocks are called monomers, and those monomers link together with dehydration reaction. Again, it doesn't matter if you're talking about how amino acids link together to form proteins, or how monosaccharides link together to form polysaccharides. It doesn't matter. They all link together the same convenient way. It's dehydration reaction. Now look at this. How would I break these bonds apart, right? So let's say I have a polymer of 1, 2, 3, 4 monomers, and I want to break this bond, and I want to release this monomer. I want to release the monomer. So I use a water molecule. A water molecule is used to attack that bond, and that frees the monomer from the rest of the polymer. You see that? And you have reconstructed this hydroxyl group and this hydrogen. So the water molecule was used to break the bond. This is known as hydrolysis, which makes perfect sense, right? Hydro meaning water, and lysis is the word meaning break. So you're using water to break the bond. Again, for example, I'll just give you an example. This might be a polysaccharide, you see? This might be 1, 2, 3, 4 sugars linked together. And I want to break this last sugar, this monosaccharide off. And I would use water to do it. Water would attack the bond, and now I have released the monosaccharide, right? This could be a monosaccharide that I broke off from the rest of the polysaccharide. Or this might be a amino acid that I broke off from the rest of the protein. Or this could be a nucleotide that I broke off of DNA. It doesn't matter. All of the biomolecules, or macromolecules, I should say, work the same way. They're built with dehydration reaction by removing a water molecule, and they're broken by hydrolysis by using water to attack. And if you're wondering, who's doing this? How is this being done? There are specific enzymes that build using dehydration reaction, and different enzymes that break bonds for hydrolysis. That's how your cells achieve this. Isn't that neat? So now let's delve into the first of the biomolecules, the carbohydrates. Carbohydrates include sugars and polymers of sugars. The simplest carbohydrates are called monosaccharides. You can think of monosaccharides as the monomers of sugars. So when I say a monomer of sugar, I mean a monosaccharide, which is a single sugar. Many sugars exist as disaccharides. I'm going to give you some examples of disaccharides as well. However, you can also form carbohydrate macromolecules as polysaccharides, which are polymers composed of many monosaccharides linked together. So what does a monosaccharide look like? Well, monosaccharides have the molecular formula that is a multiple of CH2O. So usually sugars have a molecular formula that is some multiple of CH2O. So for example, glucose is a monosaccharide. It's a very common monosaccharide. And its molecular formula is C6H12O6. So you see that? That's an example of what I mean by a multiple of CH2O. You see how there's twice as many hydrogens as there are carbons or oxygens? That's what I mean. So for example, glyceraldehyde is C3H6O3, while ribose is C5H10O5. And glucose is C6H12O6. So you see, you have all these different size sugars. But if you see a multiple of CH2O, then most likely you're dealing with a sugar. That's how you know you're dealing with a sugar. So here you can see the different sugars that I was just talking about. Here you have glyceraldehyde. Remember, this was C3H6O3. Right below it, you have the 5-carbon sugar ribose, C5H10O5. And then you have below that glucose. Glucose is C6H12O6. So you see that the thing they have in common is that they are all multiples of CH2O. And by the way, you can form, remember when we discussed isomers? There are isomers of all of these different sugars. So on the left, you can see the isomer, which is a aldose, which means a sugar with an aldehyde group called an aldose. This is glyceraldehyde. And on the right, you can see the isomer, which is ketose, which means it's a ketone sugar. It has the ketone carbonyl group. Do you remember your carbonyl groups? The aldehyde carbonyl group and the ketone carbonyl group? Well, the aldose sugars have an aldehyde carbonyl group, while the ketose sugars, or the ketone sugars, have a ketone carbonyl group. Okay, and then below that, look, you can have isomers of ribose as well. So the ribose has an aldehyde carbonyl group, whereas its isomer ribulose has a ketone carbonyl group. And look at this. All three of these are C6H12O6. So just because someone says, hey, pass me the C6H12O6, you don't necessarily know what they're talking about. Because look at this. All three of these different sugars, glucose, galactose, and fructose, they are all C6H12O6. Does that make sense? But what makes them different? Well, look at this. First of all, glucose and galactose have aldehyde carbonyl groups, whereas fructose has a ketone carbonyl group. Additionally, remember your geometric isomers? Glucose and galactose are cis-trans, right, to one another. They have cis-trans arrangements of hydroxyl groups. This one has the hydroxyl group on the right of this carbon. This one has hydroxyl group on the left of that carbon. So you remember your geometric isomers there. So again, there are so many different sugars out there. There are different length, right? Different numbers of carbons and different isomers, different orientation of those building blocks as well. Now, I want you to notice how they drew these sugars. They drew them as a linear structure. However, look what it says on your next slide here. It says that though often drawn as a linear skeleton, in aqueous solution, sugars can form rings. So let me show you what I'm talking about. Your textbook might show a glucose molecule as a linear form, which you can see on the left. However, remember when you add the sugar to water and put it in an aqueous environment, the sugar can kind of fold in on itself. It can kind of fold and form a ring structure. So take a look on the right. That's glucose as a ring structure. Take a look on the left. That's the same glucose, but in its linear form. So again, outside of solution, they adopt a linear form, and inside of solution in an aqueous environment, they form a ring structure. So just keep that in mind and don't get confused, because sometimes your textbook will show sugars looking like this and sometimes like this, but it's the same thing in reality. All right, now look at this naming here. This carbon, this is the first carbon, so this is glucose, right? Glucose has 6 carbons, and look, the first carbon is right here. How do I know that's carbon number 1? That's called the anomeric carbon. That means the first carbon. It's the only carbon attached to 2 oxygens. You see this? It's attached to this oxygen and that oxygen, right? So this is carbon 1. Next to it is carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6 is actually outside of the ring. It's up here. So this is the 6th carbon, okay? And so a lot of times these sugars are numbered, 1 through 6. If it's a 6-carbon sugar, 1 through 5, it's a 5-carbon sugar. And then for shorthand, they'll leave the carbons out. So you see this right here? They'll leave the carbons out, but you already know what to do. Remember, we discussed anywhere you see a little vertice, you'll write in your carbons. But keep in mind, each one of these little vertices are a carbon, right? And this carbon's written out because it's not part of the actual backbone. Now, remember, you can link 2 of those monosaccharides to one another to form what's called a disaccharide. Di meaning 2. Now, when they are linked together, remember that process of linking monomers together, in this case, linking monosaccharides together, is called dehydration reaction. Remember that? Dehydration reaction. And many sugars exist as disaccharides. So, for example, maltose is a disaccharide of 2 glucose molecules linked together. Lactose is a disaccharide of a glucose and a galactose. Sucrose, or table sugar as you know it, the same stuff you put in your coffee, is a disaccharide of glucose and fructose. So these are common disaccharides that just exist as disaccharides. The covalent bond that forms between 2 monosaccharides when you link them together by dehydration reaction is called a glycosidic linkage, also known as a glycosidic bond. Here, let me show you how this works. Let's go through this step by step, okay? Check out up here. I have a glucose ring structure. Right here, this is a monosaccharide. And notice how it has this hydroxyl group that's going to be important in this process. And then look, right, its next door neighbor here. This is another glucose, right? And it also has a hydroxyl group right here. The hydroxyl group from the first carbon on this glucose molecule is going to approach the hydroxyl group on the 5th carbon of this glucose molecule, you see? And do you remember how this works? Dehydration reaction. So we're going to remove an oxygen and a hydrogen from this glucose molecule, and we're going to remove simply a hydrogen from this glucose molecule as water. So this is where the dehydration comes in. And what forms when you do dehydration reaction? You form a covalent bond. This is a proper covalent bond. So notice how the oxygen from this hydroxyl functional group is left over. This carbon now bonds to that oxygen, and that oxygen bonds to the other carbon on the other glucose molecule. So this right here is the new bond. And remember what this bond is called that forms between monosaccharides? That's right, it's called a glycosidic linkage, or a glycosidic bond right here. So if I said, how do we link monomers together? Remember, that's dehydration reaction. And what's the covalent bond that forms between two sugar molecules? It's called the glycosidic linkage, or the glycosidic bond. Does that make sense? And now you have formed a disaccharide, and the disaccharide that forms between two glucose molecules is called maltose. All right? Now look below. Here we're creating sucrose. So in this case, we're going to do the same thing. We're going to do a dehydration reaction between, this time, the monosaccharides glucose and fructose. Glucose and fructose link together to form a glycosidic linkage, or glycosidic bond, forming the disaccharide sucrose. This is table sugar. But what if I want to break these sugars back apart, right? So here I have a disaccharide. This is maltose, so two glucose molecules. And I want to break them back apart into monosaccharides. How would I do that? Remember, this time I would use water. See? I use water to attack the glycosidic linkage, thereby breaking that bond, breaking the glycosidic linkage, freeing the two glucose monosaccharides from one another. And look, that water molecule is now part of these hydroxyl groups. 1O and 1H form the hydroxyl group on this glucose molecule, whereas water's other hydrogen now exists on the hydroxyl group of this glucose molecule. And do you remember what this process is called? It's called hydrolysis. And that makes sense, right? Because you're using water to break the bond. In this case, you're using water to break the glycosidic bond. Does that make sense? So let's move on to polysaccharides. Polysaccharides are polymers of sugars. You create polysaccharides both to store energy as well as for structural roles as well. Those are the two main reasons polysaccharides are formed. One type of polysaccharide is called starch. This is a polymer of glucose monomers linked together. It's a storage structure for energy reasons in plants. Plants will string glucose monomers together into long chains, and that's for use in the future. If the plant needs to use that energy from the sugar, it can then break down that starch back into glucose monomers and burn that for energy. Now, in animals, we also string monosaccharides of glucose together as well, but we don't make starch. Instead, when we string glucose together, we make glycogen. Glycogen is a polysaccharide in animals. And again, we also make this structure for energy reasons, to store that sugar energy. Let's say you eat a big meal, you have excess glucose floating around your body. Your liver and your muscle, your skeletal muscle cells, take up that glucose, and then they will do dehydration reaction to produce glycogen, to string those glucose molecules together into the polymer or polysaccharide glycogen. So here you can see the difference between the two. On the top, you can see starch. Notice how starch is, a polysaccharide of glucose. These are all glucose monomers strung together. And these form amylose, which is unbranched, and amylopectin, which is somewhat branched, but there isn't much branching going on with the glucose polysaccharides in starch. Now, look at the bottom here. This is glycogen from animals, right? From our liver and skeletal muscles. Notice this. Glycogen is, highly branched, right? Glycogen is extensively branched. And that's the big difference between starch and glycogen. You should know that starch is somewhat branched, but not very branched, while glycogen is extensively branched. But either way, they both are formed from glucose, and they both are formed to store the energy from the sugar for later. Now, what about a structural role for a polysaccharide? Well, here we talk about cellulose. Cellulose is the major component of the tough cell wall of plants. You ever heard that plants have a cell wall? Well, that cell wall is made up of cellulose, which is actually also made of glucose, but it's a different isomer of glucose. Okay, I'll show you in the next slide what I'm talking about, but this cellulose is not a storage of glucose as a polysaccharide for energy reasons. Instead, is a structural reason, right? Plants string glucose together to form cellulose as a cell wall for its structural properties because it's so tough. Now, this difference between glucose used to make starch and glucose used to make cellulose is the difference between the isomers alpha glucose and beta glucose. So here you can see it's beta glucose. It's beta glucose that's used to make cellulose. And cellulose forms the tough microfibrils of the cell walls, the plant cell walls. And you can see here what I'm talking about. Here on the left, this is glucose, but you could see the hydroxyls are cis in the cis orientation, while here in beta glucose, this is a different isomer of glucose with the hydroxyl groups and trans orientation. It's the alpha glucose that's used to make starch and glycogen for energy storage, while it's the beta glucose that's used to make cellulose for cell wall structural reasons. So you see here, when you string alpha glucose together and form a polysaccharide of alpha glucose, you see here the glycosidic bonds. And look, all of the little hydroxyl groups point downwards like this. All the hydroxyl groups point down. They're cis. But look here at the beta glucose molecule. When you string this together, when you string beta glucose together to form cellulose, notice how you end up with these staggered hydroxyl groups, right? These hydroxyl groups are now staggered, and that's responsible for the hydrogen bonding between adjacent strands of cellulose. And that's what makes the cell wall so tough. You don't get this with alpha glucose. You don't get this with glucose or glycogen. All right, so again, the alpha glucose isomer is used for formation of starch and glycogen for energy storage, while the beta glucose isomer is used to produce the polysaccharide cellulose for structural reasons. Okay, starch in its alpha configuration is largely helical. Cellulose molecules in the beta configuration are straight and unbranched. Look, starch forms helices, glycogen forms these helices, while the beta glucose, these form kind of like these straight, unbranched, and unhelical, straight, unbranched structures. Now, have you ever wondered, okay, if glucose is used to make both starch and cellulose, then how come eating starchy foods is really high in calories and you gain a lot of calories from eating starchy foods, But if you eat cellulose, if you eat, do you know what another word for cellulose is? Cellulose is also known as fiber, right? If you eat fiber, you're actually eating a lot of glucose, aren't you? You're eating a ton of glucose when you eat fiber, right? But do you gain a lot of calories from eating fiber? And the answer is no, right? So why does eating polysaccharides of alpha glucose like starch and glycogen give you so many calories, while eating the polysaccharides of beta glucose in the form of cellulose, aka fiber, not give you so many calories? Well, look at the structural difference, right? With starch and glycogen, because it's the alpha glucose variant, you have all these hydroxyl groups pointing down. And your body has the enzymes necessary to cleave this glycosidic bond, right? To hydrolyze this glycosidic bond and hydrolyze this bond and hydrolyze this bond, right? You and I, as humans, we have the enzymes necessary to break this polysaccharide down into individual monosaccharides, into glucose, and then our mitochondria can burn that glucose to produce ATP, right? So you can't get energy from this molecule unless you can break off individual glucose so that you can burn them for energy. Does that make sense? So when you eat a lot of fiber, like this structure down here, cellulose, you actually don't have the enzyme that can recognize this glycosidic bond, and so you cannot hydrolyze this bond. You can't free this monosaccharide, and that means you can't digest that monosaccharide. You can't use it for energy. You can't burn this sugar because you can't free the individual glucose molecules from one another, because your enzymes, you just don't have the correct enzyme to break this down. Right now, there are some animals that can, right? Like those ruminant animals, the ones with 4-chambered stomachs, like goats and horses and cows and things like this. These animals have special chambers in their stomachs where they can break down these glycosidic bonds from cellulose. And this is why a cow, for instance, can eat grass all day and grow to be 800 pounds, but a human would starve eating grass all day, right? Because we cannot get energy from cellulose or fiber. Isn't that interesting? So this is why they call it, insoluble fiber, and it passes through the digestive tract. It never gets taken up. It never gets burned for energy. And so you cannot make use of that cellulose you're eating, that sugar that you're eating. Now, other animals have different cell walls. So for example, many fungi have cell walls made of chitin. Chitin is another structural polysaccharide. It's found in fungi. It's found in the exoskeleton of arthropods, so insects. And you see here, it's basically a variant of glucose. You have here a glucose monosaccharide, and then you have this small functional group attached, right? So this is a monomer of the chitin cell wall, right? The chitin cell wall is just a polymer of this chitin monomer. All right, and here is a summary slide showing you the different examples and functions of different types of monosaccharides, disaccharides, polysaccharides. All right, so let's move on after a short break time with Wicket over here. I think he's done destroying things for now. I'm going to show you what he's up to right now with a special break time with just Wicket. All right, welcome back from break time with just Wicket. Let's carry on with lipids, all right? The next class of biomolecules. We're moving on to lipids. Remember, lipids are biomolecules, but are they macromolecules? That's right, Wicket. They are not macromolecules. Why? Because you cannot link multiple lipids together to form a polymer, right? There's no such thing as a polyfat, for instance, right? So because lipids do not link together to form polymers like the other macromolecules can, this is why lipids are not macromolecules, okay? So lipids. Lipids are the one class of large biological molecules that does not include true polymers. The unifying feature of lipids is that they mix poorly, if at all, with water. So they don't tend to mix with water. That's what lipids have in common. And the reason is, can you guess it? I'm going to let you think hard, all right? It's because they're mainly made of two elements, carbon and hydrogen. So when you look at these lipids, you'll see that lipids consist mostly of hydrocarbon regions, right? That means hydrogen and carbon. And do you recall from the previous couple of chapters, the important thing I needed you to remember is that hydrogen and carbon have roughly the same electronegativity, which means when you have these lipids, for instance, that are made up of almost entirely carbon and hydrogen, these are going to form nonpolar molecules. And remember, nonpolar molecules do not mix with water, right? So many of these lipids do not mix with water unless they have special functional groups attached that are polar that give them that ability to do so. Does that make sense? So the ones we're going to be touching on, the lipids we're going to go over, are the fats, the phospholipids, and the steroids. These are the three classes of lipids that we're going to discuss now. So let's start with fats, which is the first type of lipid we're going to discuss. Fats are made up of two main parts. There is a glycerol, right, and three fatty acid tails. The glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. This is what glycerol looks like, by the way. Right here, you see it's a 3-carbon alcohol with a hydroxyl group attached to each carbon. That's glycerol. And notice what it's attached to. 3 fatty acids. A fatty acid consists of a carboxyl group attached to a long carbon skeleton. So let me show you what a fatty acid is. Here, this is a fatty acid right here. This is called a fatty acid. You have a carboxyl group. See here, I'm circling the carboxyl group, followed by a long hydrocarbon tail. This is called the hydrocarbon tail. Now, in case you're wondering, why is this called a fatty acid? Well, remember that carboxyl groups are acidic. That is why this is called an acid. But what we want to do, if we want to make a fat molecule consists of a single glycerol head with 3 of these fatty acids connected to it. And the way you connect a fatty acid to a glycerol head is by dehydration reaction. You see, the hydroxyl group from the carboxyl group on the fatty acid is joined to the hydroxyl group on glycerol. 1 hydrogen from glycerol and a hydroxyl group from the fatty acid leave as water, and this results in a bond forming. See, a bond forms between that carboxyl group and the glycerol head. And now this right here, when you see O double bond C, O, C, that's known as an ester linkage. An ester linkage forms joining the fatty acid tail to the glycerol head. And you do that 3 times and you have a fat, right? So I need you to know that a fat consists of a glycerol head with 3 fatty acid tails. Now I want you to take a look at this molecule. Is it not overwhelmingly carbon and hydrogen? Right? There's so many carbon and hydrogen. So do you think that this fat molecule would mix with water? Like, do you think any partial charges are popping up anywhere? Do you think If there's a partial negative charge anywhere or a partial positive charge anywhere, well, maybe a little bit on this ester group on the tip of this O, but not enough to really make a difference, right? This fat molecule is really overwhelmingly carbon and hydrogen. And so this fat molecule is not going to stick to water. It's not going to mix with water. And this is exactly why fat and water do not mix. And now you know exactly why fat and water don't mix. Isn't that interesting? So this is known as a triacylglycerol, and it's a fat. Again, fats separate from water because water molecules hydrogen bond to each other and exclude the fats. There's nowhere for the water molecules to, interact with the fat molecule. The water molecules want to interact with anything with a positive charge or a negative charge, and there just aren't any charges on that fat molecule, right? So the oil and water separates out. Now, here's something interesting. The fatty acids in a fat can be all the same or of 2 or 3 different kinds. So what does that mean? Well, fatty acids vary in length. That means the number of carbons in the tail, and in the number and locations of double bonds. So there could be double bonds in the tail of the fatty acid. Saturated fatty acids have the maximum number of hydrogen atoms and no double bonds. This is the important part. No double bonds in the fatty acid tail. Unsaturated fatty acids have 1 or more double bonds in the fatty acid tail, so they have fewer hydrogen. So take a look here. This is a fat. And now I know what you might be wondering. You're like, what in the world is this? This does not look like a fat. But do you remember when I taught you shorthand and I told you be on the lookout because your textbook will use shorthand organic chemistry and not write everything out every time? Well, let me prove to you that this right here is a fat. It is what we were just discussing on a previous slide. So let's hop to that previous slide and hold these two up against one another so I can show you what I'm talking about. All right, here's what I was talking about. Remember on the left, this is what we said a fat molecule is, right? You've got a glycerol head with 3 fatty acid tails, okay? Now look at the right. This is the shorthand version of the same thing. Take a look. Here is your glycerol head. Here's your ester group, O double bond C, OC, see? So this right here is the analogous to this right here. Okay, and look at this. Now you might be confused by this zigzag line right here. You see this zigzag? You might be like, what is that? Charlie Brown's sweater? What is this? What's going on here? Now keep in mind, remember what I taught you before. Everywhere you see a vertice, it's a C, right? It's a carbon. And I don't know why it makes these lines across here, but everywhere you see a C or a vertice, it's a carbon, right? Remember this? So every one of these points is a carbon, remember, and you could solve for this. And so that's exactly what you see in the fat molecule. Everywhere there is a vertice, and then at the end you have a carbon at the end too. And then remember, how do we solve for the number of hydrogens? That's right. The number of bonds has to equal 4. So this carbon is making 1, 2, 3, 4 bonds. And that's why this carbon has 2 hydrogens attached. This carbon has 2 hydrogens attached. You see? So you could sit here and solve these tails, right? And do you see how, let me just clear all this. Do you see how this molecule here is the shorthand version of this molecule here? So again, don't get confused. Your textbook will show, molecules with all the carbons and hydrogens written out, or it could leave that information out and have you solve it on your own by using the shorthand. Okay, either way, don't get confused. These are both the same thing, all right? Now, hopping back to this slide, now we know that this is a fat, okay? Now, I'm going to ask you something. Look at the tails. You see these tails here? You see how there's only single bonds in the tail? See how there's only single bonds in the tail? And all 3 tails have only single bonds? Well, that's how I know it's a saturated fat, okay? A saturated fat only has single bonds in the fatty acid tails here. You see that? Now juxtapose that with the one on the right. Look at this. This fat has a saturated fatty acid tail here, a saturated fatty acid tail here. But what about this last tail? Look at this tail. What is that right there? What is that right there? That's a double bond, isn't it? That's a double bond. That tells me that this is not a saturated fatty acid tail. That's an unsaturated fatty acid tail. And even the presence of a single double bond anywhere makes this whole thing, this whole fat is called an unsaturated fat. It doesn't take more than one double bond to make a unsaturated unsaturated fat, right? So, and do you remember what happens? Let me just clear my, yeah. Do you remember what happens when carbons form a double bond? We talked about this in chapter 2. When carbon forms a double bond, or was it chapter 4? That's right, chapter 4. We talked about carbon making double bonds. Remember when carbon makes single bonds like this, it has a tetrahedral shape. But when carbon makes double bonds like this, it becomes planar, right? It changes its orientation. Those orbitals are constrained. So look, this causes the fatty acid tail to adopt a kink, right? Normal fatty acid tails, or I should say saturated fatty acid tails, are long and straight, right? But unsaturated fatty acid tails have a kink. They have a kink. All right. So what is the consequence of that kink? Well, look at this. Saturated fats, which have no double bonds in those fatty acid tails, they are solid at room temperature, like butter, lard, like, these types of animal fats, right? Animal fats tend to be solid at room temperature. And that's because of the saturated fatty acid tails. This is because when at room temperature, these straight tails can stack nicely and stop moving. And that's what freezing is. That's what a solid is. It's when the molecules stop moving. However, look at this kink here in the unsaturated fat from vegetables, right? You've got plant oils, like olive oil. Plant oils have, they're liquid at room temperature, aren't they? Why are they liquid at room temperature? Well, because of this kink, because of this double bond, the fatty acid is kinked. And when you have that kink, that means that it's harder for these fats to stop moving because they slip past each other and they stay liquid at room temperature. Isn't that neat? So what do we need to know? Again, I want you to know that saturated fats have only single bonds in their fatty acid tails, while unsaturated fats have at least one double bond in the fatty acid tail. I also want you to know that saturated fats are solid at room temperature, while unsaturated fats tend to be liquid at room temperature. Okay. I also want you to know that saturated fats come from animal products, and while unsaturated fats come from plant or fish products. Okay. And then lastly, this is the toughest one to understand. Okay. This is the toughest thing to understand. But, so what if I asked you which one has more hydrogens, a saturated fat or an unsaturated fat of the same size, right? So which one has more hydrogens? Well, think about it. Each one of these points, remember, each point is a carbon. And if it's an unsaturated fat making only single bonds, how many hydrogens does each carbon have? That's right, it has 2 hydrogens, right? Each carbon has 2 hydrogens. But look at this. When you form a double bond in the tail, how many hydrogens can these carbons have? Those double bond carbons can only have 1 hydrogen each. Does that make sense? So if I were to ask you, do saturated fats have more hydrogens, or do unsaturated fats have more hydrogens? Which one would you say has more hydrogen in the tail? That's right, the saturated fat of the same size, right? So that's something you should understand. All right, so with regard to lipids, we just finished talking about fats. Let's talk about the next class of lipids, phospholipids, and I'm going to explain to you why these phospholipids are so important. So a phospholipid is the main component of the membranes of a cell. So remember I said that all cells have a plasma membrane or a cell membrane around the cell. Cells have a membrane. And then remember the eukaryotic cells have a bunch of other membranes, you know, the membrane-bound organelles, including the nucleus, which has a double membrane. So every time you hear like membrane in a cell, like either the membrane of an organelle or the membrane of the cell itself, that membrane is usually made of phospholipid, right? Phospholipids. This is why it's so important to learn about phospholipids. They make up the membranes of your cells. So a phospholipid is made up of 2 fatty acids, a phosphate group, and a glycerol. And there's also another small piece, but we're not going to dwell on that. So here you can see this is a phospholipid right here. Notice this part here that I'm circling right here. Does that look familiar? By the way, look at this part right here. What is that? That is glycerol. Remember glycerol? That should look familiar. So we got a glycerol head. And what are these down here? What are these down here? What do these remind you of? That's right. These are fatty acid tails, right? Fatty acid tails. So look at this. You've got a glycerol head with 2 fatty acid tails. Does this look familiar? It should, because this is basically almost a fat, right? A fat is basically a glycerol head with 3 fatty acid tails, but a phospholipid is a glycerol head with 2 fatty acid tails. And then the reason it's called a phospholipid is because it has this functional group attached to the glycerol head. What is this functional group called? That's right, it's a phosphate group. It's a phosphate group. All right, so the phosphate group is attached to the glycerol head. And like I said, there's another little attachment here called choline, which is a charged entity as well. So look at this. You have a glycerol head, 2 fatty acid tails, a phosphate group. And what charge do phosphate groups have, by the way? What charge do they have? That's right, a negative charge, right? Phosphate groups have a negative charge. And then what about the choline? The choline has a positive charge. The phosphate has a negative charge. Okay, so let's take a step back. Again, what do we have? We have a glycerol head, we have 2 fatty acid tails, and then on this part, attached to the head, we have a negatively charged phosphate group and a positively charged choline group. Okay, so why is this important? Well, look at this. This molecule is very interesting. This part's called the head, okay? This part up here is called the head, and these 2 down here are called the tails. These are tails. So it's a head with 2 tails, isn't it? Now I want to show you something. This head piece is called the hydrophilic head. What does hydrophilic mean? That means mixes with water, right? Water can interact with this head, apparently. And why do you think water can associate with the head? You see this? Well, do you see these charges here? Can water interact with the negative charge on this phosphate group? That's right, Wicket. Yes, it can. And can water interact with this positive charge on this choline group? Right again, Wicket. Yes, it can. So water can interact with this negative charge on phosphate. Water can interact with this positive charge on choline. So water can interact with the hydrophilic head, right? But look down here. What are these called again down here? These are called fatty acid tails, right? In fact, the one on the left appears to be a saturated fatty acid tail, and the one on the right appears to be an unsaturated fatty acid tail. Now, what do we know about fatty acid tails? What are they comprised of? Remember, just carbon and hydrogen, actually two hydrogens, carbon and two hydrogens, carbon and two hydrogens, right? So you've got these long hydrocarbon tails. Does water interact with these long hydrocarbon tails? No, and this is why it's known as the hydrophobic tails. Does that make sense? So this is really strange. This is also known as an amphipathic molecule. This means a molecule that is part hydrophilic, which is the head, and part hydrophobic, in this case, the tails. And look how they draw in your textbook, you'll often see phospholipids drawn like this. It looks like a little head with two little tails. And you should know that the head is hydrophilic because it has charges that water can stick to, and the tails are hydrophobic because they are comprised entirely of carbon and hydrogen. All right, and you might be wondering, well, okay, but how does this form the membrane of my cells? Well, it takes millions of these to make up the membranes of your cells. So here's what it looks like. You see each one of these is a head with two tails. So what you end up with are all these heads lined up here on one side of the membrane and all these heads lined up here on the other side of the membrane. So what you have here is what's called a phospholipid bilayer. Well, it's made of phospholipids and it's a bilayer. That means two layers of phospholipids. And do you see how they line up? This would be the outside of the cell, for instance, right? So if this was the plasma membrane of the cell, this would be the outside of the cell. And what do we know is outside the cell? Liquid, right? Water, right? So there's usually solution outside of the cell, liquid outside of the cell, and that's where the heads are, because the heads can interact with the water outside of the cell. And then there's liquid inside of the cell. See, there's lots of water inside of your cells. In fact, most of your body weight is water, right? So you have lots of water inside of the cell, you have lots of water outside of the cell, and that's where the water that's interacting with the hydrophilic heads. But what about the hydrophobic tails? Look at this. They call this region the hydrophobic region of the membrane. Why? Because there should not be any water in here with these tails. These tails are just like an ocean of hydrogen and carbon, right? And there should be no water hanging out with these tails. This is the hydrophobic region of the membrane, right? So isn't that interesting? This is what your membranes look like. They are essentially a phospholipid bilayer. All right, so far with lipids, we've talked about fats, phospholipids. Let's move on to the final group of lipids, steroids. Let's talk about the steroids a little bit. Here you can see that steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. We've seen that before, right? Remember the 4 fused rings when we discussed estradiol and testosterone? Those were steroids as well. Notice the 4 fused rings that comprise the carbon backbone of those molecules, of those biomolecules. So cholesterol is a type of steroid which is a component in animal cell membranes. So animals have cholesterol in the membranes, the phospholipid bilayer of their cells. And the reason for cholesterol in our cell membranes is to keep the membrane fluid, to keep the membrane from freezing up. Now, plants do not have cholesterol in their membranes, so plant products do not contain cholesterol. Here's what cholesterol looks like. Here you can see the 4 fused rings. Here's a hydroxyl group. You've got a couple methyl functional groups, and then you have this other attachment here, this other functional group here, and this is what makes up cholesterol. All right, and here's a summary of all the key concepts we just talked about with lipids. Remember, a fat is a glycerol head with 3 fatty acid tails. A phospholipid has a glycerol head with 2 fatty acid tails. And in addition, the head has a phosphate group and choline. And a steroid has a backbone which is comprised of 4 fused carbon rings. Now let's move on to proteins. Proteins are so important. So proteins are the third class of biomolecules we're discussing. They are also considered macromolecules, right? Because they are made of building blocks called amino acids. There are 20 naturally occurring amino acids, and these amino acids string together with dehydration reaction to form the proteins. And I can't express to you guys how important proteins are. Proteins, they do so many things in the cell. Some proteins speed up chemical reactions. These are the enzymes. Other protein functions include defensive functions, storage, transport, cell communication, movement, structural support. Without proteins, cells would not grow, they would not divide, they would not function at all. Proteins are so important. You can think of them as machinery of the cell that makes the cell work. In fact, do you remember what genes are? I told you that most genes are just a blueprint for producing a particular protein. So, for example, your insulin gene, the only reason your insulin gene exists is so that your cells of your pancreas know how to make the insulin protein. It's how they know how to build that protein. Isn't that interesting? So proteins are so important. They do everything that a cell needs to do to grow, to divide, to live. Okay, proteins are not just a component of the food you eat. They are so important for, all of life's functions, right? Proteins also account for about 50% of the dry mass of most cells. Okay, and again, they do so many different things: storage, transport, defensive, enzyme, hormonal, receptors, structural, motor proteins, you name it. Proteins are involved in the cell. Now, proteins are all constructed from the same set of 20 naturally occurring amino acids. And a polypeptide, or protein, is unbranched polymer built from these amino acids. The bond between amino acids is a peptide bond. And a protein is a biologically functional molecule that consists of one or more polypeptides. So the amino acids are the monomers, or the building blocks of proteins. This is why proteins are macromolecules. It's because they are made up of amino acids, which string together to form these polypeptides, right? These proteins. Amino acids differ in their properties due to differing side chains called R groups. So here's what I'm talking about. This is a amino acid skeleton here. This is your amino acid skeleton. And what I want you to notice is this. Here on the left, you have an amino group. All amino acids have an amino functional group. Remember the amino functional group from a previous chapter. And here you have the carboxyl functional group. You see this? All amino acids have a carboxyl functional group as well. In fact, that's how amino acids got their name. Did you know that? Amino acids got their name amino because they have an amino group, and acid because they have a carboxyl group, which is acidic, right? So amino acids have amino groups and the acidic carboxyl groups as well. Every amino acid should have an amino group and a corresponding carboxyl group as well. Now, every amino acid also has a carbon in between the two called the alpha carbon, and that alpha carbon always has at least one hydrogen. Okay, so everything you see here, I'm going to put a little box up here. Everything you see here is called the backbone of the amino acid. You want to know why? Because all 20 of the amino acids share this same backbone. They all have the amino group, they all have the carboxyl group, they all have an alpha carbon with its little hydrogen, right? So this is called the backbone of the amino acid. So what is this then off to the side? Well, this is what makes the 20 amino acids different from one another, right? This is called the side chain or the R group. The R group is variable, right? That means different amino acids have different things for their R group. Let me show you what I'm talking about. Here you can see the 20 amino acids all laid out right in front of you. So take a look at this. This amino acid right here is called glycine. And notice it has the ionized form of the amino group. Remember, we learned about the ionized form of the amino group and the ionized form of the carboxyl group. You have your alpha carbon, you have your hydrogen. And so do you see why this is shaded in purple here or lilac color? Yes, that's because this is the backbone, right? What would that make this second hydrogen then? That would make it the side chain or the R group. And guess what? If you're an amino acid with the R group hydrogen, you're known as glycine. If you're the amino acid with the side chain CH3 or a methyl group as your side chain, this is alanine. It could be shortened to ALA or just A. You see, so these are the amino acids. Amino acids have a name based on what their R group is. They have a shortened abbreviation, a 3-letter abbreviation, or just a single letter abbreviation as well. So look at this. But notice how all 20 of the amino acids have that purple part of the amino acid. See the lilac part of the amino acid? Well, that's because they all share the same backbone. See? They all share the same backbone. And that's why they all have a purple part. So now why are some of the R groups, right? The R groups are in yellow, the R groups are in green, pink, and light blue. Why are the R groups 4 different colors? Let's discuss that real quick. So first of all, let's look at the yellow. Let's look at the yellow R groups. I want you to look at the yellow R groups. Notice how the yellow R groups are comprised almost entirely of carbon and hydrogen. What does that tell you about the property of these R groups, these yellow R groups? If they're comprised entirely of carbon and hydrogen mainly, that's right, Wicket. You got it. He seems like he's sleeping, but he's really paying attention. So yes, these yellow R groups are non-polar R groups. So these are known as the non-polar amino acids. When they call these non-polar amino acids, they're not talking about the backbone, which has, the charges or any. We're not looking at the backbone, okay? We're looking at the side chain. See how this side chain is entirely C and H, right? Carbon and hydrogen. So this side chain is nonpolar. So this is called a nonpolar or a hydrophobic amino acid. Does that make sense? Now look at the green ones, all right? Let's take a look here at the green side chains. What do you see on these side chains? I see a hydroxyl, sulfhydryl, hydroxyl. What does that tell me? If I have hydroxyl groups, are those nonpolar or what are those? Yep, Wicket. Exactly right. These are polar R groups, aren't they? Remember oxygen and hydrogen? Which one's more electronegative, oxygen or hydrogen? Oxygen, right? And so oxygen pulls in those electrons that are, being shared in this covalent bond, forming a polar covalent bond. That leaves hydrogen with a partial positive charge. Remember that? So remember, we learned about polarity. We learned about how to tell if molecules are polar or not. And OH is polar. Remember, like water is polar. So that means that these green side chains have partial charges on them. Okay, they have partial charges, which means they are called the polar side chains, which are hydrophilic. They will mix with water. They like to mix with water. and they like to interact with one another. Okay, next. What about these pink side chains? Well, it looks like, what is that group? What is that group at the, look at the end of these R groups. So what is this? What is this? Well, aren't those carboxyl groups at the end of these R groups? And what do we know about carboxyl groups? That's right, they're acidic. So these pink ones, aspartic acid and glutamic acid, these are acidic amino acids. And by the way, do you remember what I said in a previous chapter? A lot of times your textbook will call things an acid simply because they're negatively charged, because it's assumed that they donated protons to the solution. So when you see a negatively charged entity in the biochemistry realm, in organic chemistry, when you see negatively charged things, oftentimes they will be called acids. Again, why? Because acids donate protons to the solution, leaving them with a negative charge. Isn't that interesting? Just a fun fact for you. So anyway, these negatively charged R groups, they're acidic residues. These are acidic amino acids. Conversely, look at the light blue R groups. They have a positive charge, don't they? Because they have amino groups at the end of their R group. So this tells me that these are basic or positively charged R groups. These are the positively charged or basic amino acids. So lysine, arginine, and histidine are basic amino acids, while aspartic acid and glutamic acid are acidic amino acids. So you see, this is why the 20 amino acids are laid out like this. They have the purple part because they all share the same backbone. And then they have different colors of R group because they have non-polar in yellow, polar in green, acidic in pink, and basic in blue, right? So these are the 20 naturally occurring amino acids. And remember, these are the monomers which get linked together to form polymers of proteins, of polypeptides. Now, when you form polymers, those bonds that form are called peptide bonds. So let me show you how this works. Here I have two amino acids that I have already linked to one another. There's the peptide bond between the two. You see how this amino acid is linked to this amino acid with a peptide bond. Peptide bonds are specific to the bonds between amino acids. That's the covalent bond. And how do you make a peptide bond? Well, we'll take a look. Remember, anytime you link any kind of monomers together, these biomolecules, you do so the same way, don't you? You remove a hydroxyl group from one and a hydrogen from the next as what? As water. Remember that? That's called dehydration reaction. And that leaves you with a bond, a covalent bond. And because this is the covalent bond between amino acids, it's called a peptide bond. Now, just a quick quiz. Don't let Wiki beat you to the punch here. Here's a quiz. What were the bonds that formed between monosaccharides when you linked sugars together? When you linked monosaccharides together, what was that bond called? That's right, Wicket. Glycosidic linkage or glycosidic bond. I hope you didn't let Wicket beat you to the punch. And here, when you link amino acids together with dehydration reaction, what's the bond that forms with proteins? That's right, it's called a peptide bond. All right, and here I'm gonna give you a trick question. You ready for a trick question? You ready? All right, don't let Wicket beat you to the punch, okay? All right. What type of bonds are formed when you link different lipids to one another? Exactly. You don't link different lipids to one another. Remember, that's why lipids are not called macromolecules. They are called biomolecules, right? You don't link numerous lipids to one another and make a polylipid. There's no polyfats, right? So remember, that's why they're not considered macromolecules. So the answer is no, there's none. Now I could ask you what type of bonds form when you link fatty acid tails to the glycerol head. And remember, those were That's right, Wicket. Ester bonds. Ester bonds. Ester bonds form when you link fatty acid tails to the glycerol head of a fat, okay? But you don't link multiple fats to one another. That's not the way lipids work. Awesome. So I hope you guys are getting it. We're getting the point here. Now, let me show you something else. Do you remember what I showed you here? Every amino acid has an amino group, and a carboxyl group here on the backbone, right? Now look at this. I want you to pay attention here. When I linked these amino acids together, notice that it's the backbone that actually links together with peptide bonds, right? So the carboxyl group on this amino acid linked to the amino group on the next amino acid. See, the carboxyl group on this amino acid linked to the amino group on the next amino acid. So it's really the backbone that's being linked together with peptide bonds. You see this? Now, if this is true, let me ask you something. Doesn't that mean that every amino acid has an amino group at one end, right? It's going to have an amino group at one end. And what's at the other end? That's right, a carboxyl group at the other end. Isn't that interesting? So at one end, you're going to have an amino group hanging off, and that is actually called the amino end, or the end terminus, because it has a nitrogen. And look, what about the other end over here? That's right, this is called the carboxyl end, or the C terminus, because of this C right here, this carbon right here. Isn't that interesting? Every protein is linked together with its backbone, the lilac color part. Every protein is going to have an amino end at the beginning and a carboxyl end at the end. These amino acids are linked together with peptide bonds. So it's really the backbone that's strung together. And what's poking out the sides? What's poking out the sides? The side chains. That's why they got their name. Side chains poke out the sides. Remember, the side chains are also called the R groups, or the variable R groups. Yellow ones were nonpolar, green ones were polar, the pink ones were acidic, and the blue ones were basic. So that's essentially what a protein is, you guys. A protein is actually a long string of these amino acids linked together in a long string. Like, think of if each of these amino acids is like a pearl, like a pearl, then every protein is like a long pearl necklace, isn't it? Every protein is like a pearl necklace, and each pearl would represent one of these amino acids. Isn't that interesting? Okay, so every protein is simply a long chain of amino acids that were linked together with peptide bonds. Now, that's not to say that proteins stay a long pearl necklace. So proteins are essentially a long pearl necklace of amino acids all strung together. But what you need to realize is proteins fold up. They don't stay long, slender structures. They fold up into these three-dimensional structures, right? So proteins fold, and they fold into a very specific three-dimensional architecture. So here you can see this is a protein. See, so you might be looking at this, and I'll say, hey, this is a typical protein right here. And you might say, but Dr. D, that doesn't look anything like a string or a pearl necklace. It looks like a ball or a glob of something, right? So this is the folded protein. That's why it does not look like a pearl necklace, okay? The red dots are oxygen, the gray dots are carbon, and the blue dots are nitrogen, right? And you can see here, this is the space-filling model showing you where all the atoms are, except the hydrogens, of the protein. But if I were to have X-ray vision and just look at this pearl necklace, what if I could just trace the pearl necklace? Well, then look at this. Here is, actually, I'll put them side by side for you. Look at this. This is the protein's space filling model on the left, And on the right, you can see what's called its ribbon model or ribbon diagram. And notice that it really is a long chain. In fact, look, this appears to be one end right here, right where my mouse is. That's one end of the protein. And here, that appears to be the other end of the protein. You see that? So it's essentially one long chain. I could follow it. See, I could follow it here. And then look what it does. It starts coiling up into a ribbon, into a coil, right? That's called an alpha helix, by the way. You see this little twirling right here? That's an alpha helix. And then I could follow it all the way down to the other end. So yes, if I grabbed the protein from this spot right here, and if I grabbed it from that spot right there, I could theoretically pull it back into a long string like a pearl necklace. Isn't that neat? So proteins, a lot of times your protein will show you a picture of a protein as kind of a blob. But remember, at its core, it really is a pearl necklace made up of a long chain, a single long chain of amino acids. Isn't that interesting? Now, when we're talking about protein structure, there is 4 levels to protein structure. Let's go through each of the 4 levels of protein structure. When we're talking about a protein's primary structure, it's not even a real structure. It's simply a list of the amino acids in order. So remember I said, a protein is simply a pearl necklace of amino acids in a particular order. Well, if I were to list out that order from beginning to end, that's the primary structure of the protein. So here you can see the primary structure of this. The primary structure of this protein starts with the amino acid glycine, then proline, then threonine, then glycine, then threonine, then glycine, right? So it goes, this is the primary structure. It's like all the amino acids in order are the primary structure of the protein. Okay, so what's secondary structure? Secondary structure is found in most proteins, and it consists of 2 main shapes that form from the pearl necklace. The pearl necklace makes 2 main shapes: coils, right, called alpha helices, and folds, which are called beta sheets. So let me show you what those look like. Remember, there were these coils that I showed you, these coils. Those are called alpha helices. And then the folds, or beta pleated sheets, beta sheets for short. Let me show you what I'm talking about. Remember, you had this protein right here, and it has this globular structure to it. But if we were, like Superman, and we could zoom in on that, look at this. So what I would say is, look at this. See where it spirals here? That's an alpha helix. See where it spirals here? That's an alpha helix. Here's a little alpha helix. Here's an alpha helix. But where are the beta pleated sheets? Can you spot a beta pleated sheet? Maybe Gizmo can spot a beta pleated sheet. Good job, Gizmo. That's it right there. And actually, there's one right next to it, too. Look at this. See how it's a fold which is kind of flat, right? This flat region is a beta pleated sheet. And see this flat region here, this fold that's flat, that's also a beta pleated sheet. So there are 2 main structures in the secondary structure, right? There are these alpha helices and the beta sheets. And by the way, these are called loops. You see where it kind of loops over, these little loops. So you've got loops, you've got beta sheets, and you've got alpha helices. And those are the common secondary structure shapes of a protein. In fact, if I were to break down this protein, I don't know what protein this is, but I would say this is a protein that consists of 1, 2, 3, 4, maybe 5, 6, 7 alpha helices, and it looks like 2 beta sheets, right? So if I were to break down the secondary structure of this protein, 6 or 7 alpha helices and 2 beta sheets, right? Isn't that interesting? So, and you'll notice over time, when you look at more and more proteins, especially if you look at the ribbon model of those proteins, you will notice that, a lot of proteins have, these alpha helices to them. They have alpha helices or they have beta sheets, and you'll notice that just it's very common for proteins to have these two types of secondary structures. But then those secondary structures, did you notice how those alpha helices and beta sheets kind of folded in on themselves and formed a proper three-dimensional overall shape? That's the final folded protein. That's called the tertiary structure of the protein. The tertiary structure is determined by interactions among the various side chains, or the R groups. Isn't that interesting? So let's go back to our little diagram here. So again, the secondary structure were all these little alpha helices and beta sheets, but do you see how they're all kind of coming together to form this three-dimensional structure? Well, that's the tertiary structure of the protein, and that's such an important structure. I have a saying, it's that if a protein does not fold correctly, it won't function correctly, right? So a protein's fold is so important to its function. When a protein becomes misfolded, oftentimes it loses its function. It can't do its job anymore, okay? So proteins need to fold a very specific way. And you see all these secondary structures came together for this protein to fold exactly in this way so that it looks exactly like this, right? That's called the tertiary structure, or the final folded protein structure. And inside of here is one long pearl necklace, one polypeptide chain, right? And again, I don't know if this end right here, you see this end right here? I don't know if that's the N terminus or the C terminus, but I know it's one of the two, right? I don't know which one it is. But if this is the N terminus, then the other one would be the C terminus. Remember that? So it really is one long polypeptide chain. It really is one long pearl necklace in here. Consisting of a primary structure, which is simply a list of the amino acids in order, and proteins can be hundreds or thousands of amino acids long. And then the secondary structure, which comprised of alpha helices, beta sheets, and these loop structures. And then those fold in on one another to form the tertiary structure, the final folded protein, which is necessary for its function. And do you remember what it said? What's responsible for this tertiary structure? What's responsible for keeping the, for the final folded structure of a protein? That's right, the side chain interactions. So interactions between the R groups. I'm going to explain this next. All right, so I went and I snagged this pearl necklace from my wife, and I want to show you what I'm talking about a little bit here. So again, a protein is essentially a long chain of amino acids, right? And the primary structure is simply the name of the, if you list the amino acids in order from beginning to end, that's the primary structure. And then remember, parts of it will coil up into little alpha helices, these coils. And they'll also fold into these straight folds called beta sheets, right? Beta sheets as well. But now I want to show you how the whole thing folds up into the tertiary structure, into the ball. Now remember, each of these pearls represents an amino acid, right? And each one has a different charge on the R group. Remember the side chain that points up? The side chains have different charges. Remember, the yellow ones were nonpolar, the green ones were polar, the pink ones were acidic or negatively charged, and the blue ones were basic or positively charged. So let me ask you this. What if this part of the protein met this part and these two pearls met? You see, these two pearls come together at some point. You see? Well, think about it. If this is a positively charged basic amino acid, R group, and this is a negatively charged acidic one, so then a plus charge comes together with a minus charge. Could they interact and bond to one another with their R groups? Would their R groups bond to one another? Again, this one's basic or positively charged. This one's acidic, negatively charged. Would those R groups bond to one another? Yes, they would. And what kind of bond would that be? A positive charge bonding to a negative charge. Here, this is exactly what I'm talking about here. Look, right here, you've got one pearl with its R group that's acidic, negatively charged, and then another pearl with its R group that's basic, positively charged. When those two R groups come into contact, when those two pearls come into contact, they will form an ionic bond. So ionic bonds form between acidic and basic residues in the tertiary structure, right? In the protein structure. And that's partly responsible for the proper tertiary bond formation and the tertiary structure of the protein. Now, remember the pearls that were neutral? Some pearls have no charge, right? So what happens if a neutral pearl meets another neutral pearl, a non-polar pearl meets another non-polar pearl? Non-polar substances can interact as well. These are called hydrophobic interactions, right? So hydrophobic interactions occur between non-polar R groups. They like to stick to one another. What about the polar R groups? Remember the green polar R groups? Yes, polar R groups like to stick to other polar R groups and form what kind of bonds? What does this denote? Little series of little dots. What did that mean? That's right, Wicket. Those mean hydrogen bonds, right? Tiny dots in a row denote a hydrogen bond. There's a little bit of stickiness between this partially positively charged hydroxyl group on this R group and this partially negatively charged oxygen on this R group. You see? And then lastly, so again, the tertiary structure of the protein is made thanks to all these R group interactions. And there are ionic bonds that form, hydrophobic interactions that form, and hydrogen bonds that form. But there's one last one that I want you to know about. And this is really cool, okay? This is a disulfide bridge. Have you heard that term before? Do you remember when we discussed what is a disulfide bridge? When does a disulfide bridge form? Let me show you. Can you answer before Wicket? When does a disulfide bridge form? Yep, between sulfhydryl groups. I want you to take a look here at the amino acids. There's 20 naturally occurring amino acids, and there's only one that has a sulfhydryl group at the end of the R group. You see this? This is the amino acid cysteine. We talked about it a little bit in the previous chapter. Cysteine's R group has a sulfhydryl functional group at the end, sulfhydryl. And do you remember what I said when we talked about functional groups in the past? When one sulfhydryl group meets another sulfhydryl group, what happens? They form a real covalent bond known as a disulfide bridge. So guess what? If your pearl necklace has a cysteine residue over here and a cysteine residue over there, if they were to come into contact with their R groups, those sulfhydryl groups would interact and they would form a disulfide bond, right? Isn't that neat? So let me show you. Yeah, see? So this would be the sulfhydryl group from this amino acid and the sulfhydryl group from that cysteine on that amino acid, right? So there you go. This would form a disulfide bridge. Isn't that neat? So if I were to ask you, let me say, if I were to ask you, what are the 4 types of bonds that form that help to create the tertiary structure of a protein? What would you say? Well, you would say those are interactions between the R groups, and those interactions include ionic bonds, hydrophobic interactions, hydrogen bonds, and disulfide bridges. And the disulfide bridges are the rarest ones, right? They only form between cysteine residues, cysteines that meet. Isn't that interesting? So let me show you one more thing that's really interesting. Do you remember this tertiary structure? Just for fun, I want to show you something. Do you see how there are red oxygens, gray carbons, and blue nitrogens? That makes sense. But do you see the yellow atoms as well? Here's a yellow atom. There's a yellow atom. There's a yellow atom. These yellow atoms represent sulfur, and usually those are the sulfurs of the cysteine residues. So when you see a ribbon diagram like this, and you see these yellow lines like that, those are the disulfide bridges. Those are the covalent bonds between cysteines. Isn't that neat? So from now on, when you see these ribbons, you're not confused. You know that this is a protein. You know the coils are alpha helices and the folds are the beta sheets. You know that the hard yellow lines are the disulfide bonds between cysteine residues. You know that there should be two ends to the protein. One is going to be the N terminus and the other the C terminus. And this proper folding is important, right? The tertiary structure of the protein is important for the protein's function. Isn't that cool? So then you might be wondering, well, then what in the world is a quaternary structure? I thought you said a tertiary structure is the final folded protein. Well, how much more final can you get? What's quaternary structure then? Well, that's a good question. A quaternary structure results when a protein consists of multiple polypeptide chains. Some proteins don't function unless you have two polypeptide chains. You can think of it as two tertiary structures working together to make a functional unit. That's a quaternary structure. But most proteins don't form a quaternary structure. Most proteins work all by themselves. They don't need a friend to work properly. So only when two or more proteins must work together in order to function is that a quaternary structure. So here are some examples of quaternary structures. Again, quaternary structures result when two or more polypeptide chains form a macromolecule. Collagen is an example. Collagen is a fibrous protein consisting of 3 polypeptides coiled like a rope. And here's its structure. You see you have collagen, which is made up of the red protein, the blue protein, and the green protein, all kind of coiled up together. And they all form alpha helices. But this would be a quaternary structure because it is made up of 3 proteins. Next, hemoglobin is another very common example of a quaternary structure. You've heard of hemoglobin, right? A globular protein consisting of 4 polypeptides. This is the protein in your blood, your red blood cells, that helps you to transport oxygen to your cells, right? To transport oxygen around your body. Hemoglobin. Hemoglobin is made up of 4 individual polypeptides. 2 alpha proteins and 2 beta proteins. These are known as the alpha and beta subunits. Here you go. You can see here, this is hemoglobin right here. This is hemoglobin. This is the common protein that your red blood cells are full of these hemoglobin quaternary structures. You've got, here you can see 2 beta subunits, 2 beta proteins, and 2 alpha subunits, 2 alpha proteins. And notice that you also have these hemes, this iron. You know when they say you have iron in your blood, right? That's literally the element iron, or Fe, is in the blood, right? It's part of the hemoglobin. It's important for transporting oxygen around. And just to make clear, I want to show you an example of what I mean by when I say proteins must fold correctly in order to function correctly. I'm going to give you a nice example here. And the example has to do with a human disease known as sickle cell anemia or sickle cell disease. And this sickle cell disease is a disease in which instead of someone's red blood cells looking like this is what a typical red blood cell looks like. It looks kind of like a coffee bean shaped. Here, people with sickle cell disease or sickle cell anemia, their red blood cells actually look like a crescent moon, or the sickle, the farm instrument, the sickle. You can think of the Grim Reaper, you know how he carries his sickle around. This is how it got its name, because it looks like that farm instrument, the sickle. So you might be wondering, how come people with sickle cell anemia don't have red blood cells like most people like this? You know, their red blood cells have all stretched out into this curve, right? What if I told you it has everything to do with the tertiary structure? It has everything to do with the protein's structure. Isn't that interesting? So let me explain. So here you can see the primary structure of the normal beta subunit of hemoglobin. This beta subunit has these amino acids in order, right? Remember, that's the primary structure. The first amino acid is valine, followed by histidine, followed by leucine, then threonine, then proline, then glutamic acid, right? Okay, now notice when you have the proper primary structure, the proper list of amino acids in the proper order, this results in the pearl necklace folding correctly into the properly folded beta subunit. This is the tertiary structure of the beta subunit of hemoglobin. It folded correctly, and when it folds correctly, it partners up correctly with its partners. 2 alpha subunits, 2 beta subunits all come together as a, remember, quaternary structure called hemoglobin. And hemoglobin's job is to float around in your red blood cells. Your red blood cells would be full of hundreds and hundreds and hundreds of these hemoglobin proteins. And these hemoglobin proteins carry oxygen around your body, right? And that's how it normally works. Okay, and these proteins do not associate with each other. Each one carries oxygen, and each one is a separate little entity. You see that? Each quaternary structure just floats about, and they're all floating around inside of the red blood cell. So imagine if I were to cut this red blood cell open, hundreds and thousands of these would pour out, right? These hemoglobins would just pour out of the cell. Does that make sense? But now let me show you the difference between that and sickle cell. So people who suffer with sickle cell disease, their beta hemoglobin gene has a mutation. Remember what we said earlier, that genes are the blueprints for proteins? Genes code for proteins. Well, this is the protein for the beta hemoglobin, right? There's a gene that codes for this. And if you inherit a gene with a mistake, that means a mutation, that could change the blueprint for beta hemoglobin. See, so instead of having glutamic acid as the 6th residue, you see, the 6th amino acid in your pearl necklace should have been glue or glutamic acid. Now these patients have a valine amino acid as their 6th amino acid in their pearl necklace, so they have a mistake. The 6th amino acid should be what? The 6th amino acid of beta hemoglobin should be glue or glutamic acid, which means it's one of the pink amino acids, right? It's an acidic, negatively charged amino acid, okay? And what did it get changed to? Do you remember what they changed it to in the sickle cell mutation? Changed that to a V, or valine, V. Valine is a non-polar amino acid. Now think about the problem that would cause, right? Remember my pearl necklace analogy here? Let me show you. So imagine if this is, an acidic glutamic acid, and it normally pairs up with a basic amino acid to form an ionic bond to form the proper tertiary structure of beta hemoglobin, to form the proper folding hemoglobin. But what if I change that from a negatively charged amino acid to a nonpolar amino acid? Would it still pair up the right way with its basic counterpart, or would it repel? It would repel, right? So imagine, instead of a negatively charged amino acid, you now have a nonpolar amino acid. So that's not good because remember, proteins need to fold correctly in order to function correctly, right? And look at this. So again, imagine we just swapped a acidic, you see this would be an acidic amino acid with a negative charge that's supposed to make an ionic bond with a positive charge. What if we swapped this out with valine, which is, kind of like this, like nonpolar? Would it still want to hang out here with its basic friend and form this ionic bond? No, it would repel. It would push itself apart, right? And that would affect the protein structure. That would affect the nicely folded protein. And that's exactly what happens because these people have a gene mutation, which results in a valine in the 6th residue. That results in what's called sickle-cell beta subunit, which does not fold correctly, right? This is the properly folded beta subunit. This is the misfolded beta subunit. And look what happens. They still come together and form the quaternary structure with the other subunits, right? However, look what they have. You see this little opening here? You see this little groove right here? That shouldn't exist. You see there's no groove here. You see this little groove? Well, that's an exposed region, right? That's an exposed area that's hydrophobic, right? Hydrophobic. That means this part of the protein does not like water, okay? Do you see a problem with that? What is the cell full of? The cell is full of water, right? What is this part of the hemoglobin not like? That's right, it doesn't like water. So look what happens. These proteins, these hemoglobin quaternary structures, they don't like water. And so what they do is they stick to each other to squeeze the water out, right? So because those hydrophobic regions formed, the hemoglobin quaternary structures all stick to one another, forming these long fibers, these aggregates, these protein aggregates, these fibers. And the fibers literally stretch. And imagine these fibers are forming inside of the red blood cell, and those fibers are literally stretching the red blood cell out. Isn't that interesting? Should hemoglobins form fibers? Should normal hemoglobin quaternary structures form fibers? No, they just kind of float around and mind their own business, right? So a red blood cell has these quaternary structures inside, and they're all just kind of floating around, right? But because of the misfolding, this big hydrophobic region, this exposed hydrophobic region, does not like water, so they all kind of end up sticking to one another, forming these long fibers stretching out the cell. Isn't that interesting? So this just highlights for you the importance of understanding quaternary structure and tertiary structure and protein structure, because without doing so, you can't understand normal human diseases such as sickle cell. Isn't that neat? Okay, one more thing I would need you to know about proteins is that physical and chemical conditions can affect the structure of the protein. Think of the protein as this beautifully folded, delicate, three-dimensional structure, right? The tertiary structure. And remember, how are those proteins folded? What's responsible for the tertiary structure of a protein? Remember, it was hydrophobic interactions, it was ionic bonds, it was sulfhydryl groups, meeting and making disulfide bridges, and it was these polar groups meeting and making hydrogen bonds, right? So these are most of them are relatively weak interactions. So what's holding a protein in its three-dimensional tertiary structure
a lot of those interactions are kind of weak, right? So what do you think would happen if I took my nicely folded protein and I boiled it, or I placed it in acid, or I placed it in a very salty environment, right? Well, that would cause these delicate interactions to break, right? And so my protein might unfold. You see what I'm saying? If I boiled my protein, it might start unfolding a little bit, right? And there's a term for this where environmental conditions or physical or chemical conditions such as pH, salt concentration, temperature, and other environmental factors can cause my protein to unravel. This means my protein unfolds. It doesn't mean my protein falls apart into little pieces. It just means it loses its nice, perfect little tertiary structure, right? It unfolds. And this is called denaturation. A protein is denatured when it unfolds, right? And what you need to know is that once denatured, a protein is biologically inactive, right? So denaturation is not usually reversible. Denaturation can sometimes be reversed when the denaturing agent is removed. However, usually once a protein is denatured, it will not renature. So once a protein, see here on the left, you have the normal protein with its nice tertiary structure, that's nicely folded structure. You can see all the little alpha helices and beta sheets and such. Yeah. Well, what if I expose this protein to, those environmental factors or those chemical factors? You see, like pH or extremes or temperature. Well, then I can cause my protein to denatured, right? And it becomes denatured. And that means my protein has lost function. Does that make sense? When a protein denatures, it loses function. And once a protein denatures, it does not typically renature. So, there's a saying that you can't unfry an egg, right? When you put the egg on the skillet and you heat it up and the egg, turns all from like a transparent to like a white color, you can't just cool it back down and have it go back the way it was, right? Because those egg proteins are denatured, right? And usually proteins, once denatured, they will not renature. And so you could destroy the function of a protein by causing it to denature, by changing the environment drastically. Okay, and this leads us to our summary slide for proteins. Before we jump into our final biomolecule, the nucleic acids, let's take a quick break time, this time just with Gizmo, and see what that little guy's up to. And we'll be right back for the final biomolecule. All right, welcome back from break time with just Gizmo this time. Finally, we've reached the fourth biomolecule, which also happens to be a macromolecule. These are the nucleic acids, DNA and RNA. Now remember, the amino acid sequence of a polypeptide or protein is programmed by a unit of inheritance called a gene. I've been mentioning this. A gene is a unit of information for the blueprints of a protein, right? Your cells know how to make the correct proteins by following the instructions on a gene. Genes consist of DNA, a nucleic acid. In fact, DNA stands for deoxyribonucleic acid. These nucleic acids are made up of monomers or building blocks called nucleotides. Both DNA and RNA are nucleic acids made up of nucleotides. So let's talk about nucleotides. Again, there are two types of nucleic acids: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Remember we touched very briefly on gene expression in chapter 1? We talked about the lens of the eye. So the gene codes for a protein, right? The gene codes for a protein. But remember, there's that intermediary step. We're going to learn all about this later this semester. But this process of a gene resulting in the production of a protein is called gene expression. And first, you start with the gene. You copy the gene information into RNA called messenger RNA. And then that messenger RNA is translated into a protein. So whenever you see messenger RNA or mRNA, think of an RNA copy of a gene. It's an intermediary step during gene expression. And we're going to go into this in so much detail later on, so don't worry about gene expression right now. But here's the process of gene expression. You have your double-stranded DNA. DNA chromosomes include genes. The genes are the blueprint or for the proteins. We got to make a copy of that gene as a single-stranded mRNA. That mRNA is then translated by your ribosomes into that, remember that pearl necklace, the polypeptide chain, to make the protein, right? So this whole process of copying a gene to mRNA and then translating that mRNA's message to protein is called gene expression. And so in this part of the chapter right now, we're going to talk about what does DNA look like? You know, how is it built? And what does RNA look like? And how is that built? Remember, they are made up of monomers or building blocks called nucleotides. Here you can see nucleic acids are simply polymers called polynucleotides. They are polymers of nucleotides. Nucleotides are the building blocks of DNA and RNA. Each nucleotide consists of 3 parts, okay? A nitrogenous base, a pentose sugar. Pentose means 5 carbon, by the way. A pentose sugar, and 1 or more phosphate groups. So these are the 3 parts of a nucleotide. So let's focus in on a nucleotide real quick. I want to show you what a nucleotide looks like right here. Let's zoom in on this. This right here, what you're looking at in the center, is a nucleotide. Remember, a nucleotide is made up of this mustard colored region. This is the nitrogenous base. Then you have this part down here in green. This is a pentose sugar. That means a 5 carbon sugar. And then this part here, remember, what is this? You should know this part. I'm going to let you guess. That's right, Wicket. That is a phosphate group. These are the building blocks for DNA and RNA. They have a nitrogenous base, they have a pentose sugar, and they have a phosphate group. Okay, now let me break this down a little more. Ready to go into a little more depth? Here's a few things to understand. First of all nucleotides have, at least 1 phosphate group attached. Do you see this phosphate group attached? And the phosphate group is the same regardless of the nucleotide you're talking about. All nucleotides have the same phosphate group, the same structure here. But now let's look at the sugar real quick. You see this pentose sugar? Remember, pentose sugar means a 5 carbon sugar. Now this does vary depending on whether you're talking about DNA or RNA. I just want to show you something. Look right here. You see this pentose sugar, this 5 carbon sugar? Well, in DNA nucleotides that make DNA, that sugar, this is the sugar that lives right there. This sugar is called deoxyribose. Okay, deoxyribose. Why is that important? Well, because this sugar here, for RNA, this sugar is actually a slightly different pentose sugar. It's called ribose. You see this? Do you guys remember ribose earlier this chapter? Ribose is C5H10O5, right? So you see why it's called the pentose sugar, because it has 5 carbons. Okay. Now, what is the difference? Look closely. What is the difference between ribose, which has a hydroxyl group here, and deoxyribose, which just has a hydrogen here? It's missing that oxygen, right? Deoxyribose is missing that oxygen. So technically, did you know deoxyribose is technically C5H10O4? That's right, O4, because it's missing one of its oxygens. And that's why it's called deoxy, so without an oxy. It's missing an oxygen. So you see on the second carbon, remember the 2'? This is called the 2' carbon. Remember we talked about this a little bit earlier. The first carbon of the sugar is the anomeric carbon, which is the only carbon connected to 2 oxygens. Then you have the 2' carbon next to it, 3' carbon, 4' carbon, and remember the 5th carbon, the last carbon's not part of the ring, right? So anyway, the second carbon, or the 2' carbon of ribose, has a hydroxyl group attached, doesn't it? However, the 2' carbon of deoxyribose does not have that oxygen. It just has the hydrogen. And that's why it's called deoxyribose. Isn't that neat? So the reason why DNA is called DNA is because it's missing that oxygen on ribose. It's deoxyribose. And that's why ribose, is the sugar in RNA, because it's called RNA, ribonucleic acid, because it has a proper ribose sugar. Isn't that neat? So again, just to recap, nucleotides are the basic monomers of DNA and RNA. They all have at least one phosphate group attached to it. They have a pentose sugar. Now, just to quiz you, what's the pentose sugar of RNA nucleotides? That's right, Wicket. It's ribose. And what's the pentose sugar of DNA nucleotides? I hope you didn't let Wicket beat you, because you're right, Wicket, it is deoxyribose, okay? So far we understand the phosphate group, which is negatively charged. We understand the pentose sugar, which is either ribose for RNA or deoxyribose for DNA. Now, what about this part here, this mustard-colored part here? This is where it gets a little more tricky. This is called the nitrogenous base, okay? This is the part of the nucleotide that gives the nucleotide a code, right? You've heard of the genetic code, right? You guys have heard of the genetic code? The genetic code A, G, C, T. That's the alphabet of DNA, right? And RNA has a slight variation. Instead of T's, it uses U's, right? So this is the part of DNA that gives you your code, the nitrogenous base, with all the different types of nitrogenous bases. So I want you to pay attention right here. You see what I'm circling right here? I'm going to be talking about all the possible iterations of what you can have right here as a nitrogenous base, right? So just focus right here. These are the possible nitrogenous bases. You see this? Take a look here. These are the possible nitrogenous bases. So when I say nitrogenous base, I'm talking about this, or this. These are the 5 nitrogenous bases that can exist in that mustard-colored part, the third part of the nucleotide, right? And remember, I told you that DNA has a code. It can have a C, see? C, no pun intended. C for cytosine. That's this one ring structure. DNA can have T's for thymine, which is this one ring structure here. DNA can have A's. A's stand for adenine. That's this nitrogenous base here. And notice how this nitrogenous base has 2 rings. And then it can also have a G, so guanine. That's this 2-ring structure here. So just to summarize, DNA nucleotides can have these 4 nitrogenous bases. They can either have a C, a T, an A, or a G. Isn't that interesting? And for RNA, it also has nucleotides. But what's the exception I told you to remember? That's right. Instead of T's, RNA utilizes U's. U for uracil. Okay, so RNA would have C's, U's, A's, and G's. DNA has C's, T's, A's, and G's. Okay, now do you notice that some of these have one ring and some of them have two rings? So that's actually important to remember. I want you to remember that the one ring nitrogenous bases, the C's, T's, and U's, these are called the pyrimidines. You see the pyrimidines. Whereas the two ring nitrogenous bases, right, A and G, they're called the purines. So I may ask you, which ones are the pyrimidines and which ones are the purines? Do you want to know how I remember it? You know how I have silly ways of remembering things, right? And this one's silly. So bear with me, buckle up, because I'm going to take you on a mental journey to remembering the pyrimidines and the purines. So to remember this, I have this silly saying, think of a pyramid, Now, would you agree that pyramids have sharp edges that, and would you agree that a single pyramid with its sharp edges could cut you? Okay, well, if you agree with all that, you should just remember that it takes a single pyramid to cut you. One pyramid can cut. And why do I say all that? Well, because the one ring, look at this part, the one ring nitrogenous bases are the pyrimidines, which sounds like pyramids, okay? And it only takes one pyramid to cut you, right? So that's a one ring pyrimidine. And cut includes the letters C, U, and T, right? So one pyramid can cut. The single ring pyrimidines include C, U, and T. Anyway, like I said, it's silly, but it's to this day it helps me. The one ring's cut. It's cut. I just remember cut. So the single ring pyrimidines include C's, U's, and T's. And of course, whatever's left over, the A's and the G's, are the purines. Isn't that interesting? So that's how I remember all of the material. Again, just to summarize, this is a nucleotide. Nucleotides make up the DNA and the RNA, right? They are the monomers, the building blocks of DNA and RNA. Both the DNA and RNA nucleotides have phosphate groups, which are negatively charged. However, only the DNA nucleotides have the pentose sugar called deoxyribose, which is missing an oxygen on the 2' carbon. But it's the RNA nucleotides that have ribose, which does have a hydroxyl group on the 2' carbon. However, where the differences are more pronounced is in the nitrogenous base. Remember this? Oh, by the way, I only see one ring here. All right, can you beat Wicket? I only see one ring here as the nitrogenous base on this example. So which nitrogenous bases must this be? That's right, it's either a C, U, or T. I know that this is not an A or a G because A's and G's have two rings. Okay, so isn't that interesting? Anyway, the nitrogenous bases for DNA nucleotides could be A, G, C, or T. Whereas for RNA nucleotides, we have A, G, C, and U, right? In the place of T's, RNA utilizes U, or uracil. Isn't that interesting? So this is good to understand, all right? Now, there's something else to learn, okay? We got more to learn. Hopefully now you understand the nucleotide. Now I need to explain a few more things to you, okay? Here it is. This is important. This is important. You see the phosphate group right here? You see the phosphate group right here? Which carbon on this sugar, which carbon is it attached to? It's not attached to the first carbon, the 1' carbon, 2', 3', 4', 5'. So the phosphate group is connected to the 5th carbon of the pentose sugar. Does that make sense? This is the 5' carbon, which means the 5th carbon of the sugar, the pentose sugar. And the phosphate group is connected to that 5' carbon. So you know what? I need you to remember that, because a lot of times scientists will call this the 5' phosphate group, and that's what they mean, right? When they refer to this phosphate group of nucleotides, they often call it the 5' phosphate, okay? And that's why, because it's the 5th carbon of the pentose sugar that's connected to that phosphate group. Now, here's one more thing I need you to know. One more thing I really need you to know, and that is this carbon right here. This is called the 3' carbon, okay? So you can see here, the 3' carbon has a hydroxyl group attached. You see the 3' carbon always has a hydroxyl. The 3' carbon has a hydroxyl group attached. Now you might be thinking, Dr. D, why do I need to remember that? Well, because this is so important for how these nucleotides link together. You know, nucleotides they're monomers, right? This is a monomer. These monomers need to link to one another. Does that make sense? Monomers need to link to one another. So how do the monomers link to one another? Well, here's how. The 5' phosphate of one nucleotide, you see this part of it, if I had 2 nucleotides, okay, if I had 2 of these nucleotides, the 5' phosphate of one nucleotide would connect to the 3' carbon on the next nucleotide, right? Does that make sense? And yes, this hydroxyl would end up leaving in a dehydration reaction. Remember, the OH leaves in a dehydration reaction? So what you end up with is the 5' phosphate of one nucleotide connects to the 3' carbon on the next nucleotide via a dehydration reaction. And that's how you start making a strand of DNA or a strand of RNA. Let me show you a strand of DNA or RNA. Look. So look at this on the left right here. Look at this. This is called a strand, okay? Have you ever heard of a strand of DNA or a strand of RNA? This is exactly what they mean by a strand, right? This is a single strand of DNA. Or it could be a single strand of RNA. I don't know which one they've picked here, but you know, it doesn't really matter. They're very similar. And so do you see what these structures are on the left, right? What is the little yellow ball right here? What does the little yellow ball represent? It represents this phosphate group, right? Do you see this? The phosphate group. What does this green 5 carbon structure represent? This green structure. What does that green structure represent? It represents the pentose sugar. And what do these mustard colored 1-ring or 2-ring structures represent? That's right. They represent the 1-ring ones are the C's, U's, and T's, and the 2 rings are the purines, right? The purines, right. So how many total nucleotides make up this strand right here? How many total nucleotides make up this strand depicted on the left? That's right, Wicket. Star student here, Wicket. 4 nucleotides, okay? We have 1, 2, 3, 4 nucleotides all strung together in a strand of DNA or RNA. And let's see how it works real quick. Let's zoom in. Let's zoom in real quick. And I just want to show you something. So here is a nucleotide, right? You've got the nitrogenous base, you've got the pentose sugar, and you've got the phosphate group. And what did I say? This is called the 5' phosphate, right? And how does the 5' phosphate connect to the next nucleotide, this second nucleotide up here? Where did I say the 5' phosphate connects to the next nucleotide? What did I say? Remember I said it's the 1, 2, 3' carbon. You see this right here? right here, this is the 3' carbon on this nucleotide up here, right? And notice what happened. Like I said, via a dehydration reaction, the phosphate group from this nucleotide connects with a covalent bond to the 3' carbon on the next nucleotide. See, that's how I said they connect to one another, right? And then how do you think this phosphate, this 5' phosphate, connects to the next nucleotide? Yes, it connects to the 3' carbon. And look at this 5' phosphate. It connects to the next 3' carbon. So you see, that's how nucleotides connect to one another. Isn't that neat? So if this is how they connect to one another, doesn't that mean that you're always going to have a phosphate group hanging off at this end, right? Does that not mean that? Think about it. Does that not mean you're always going to have a phosphate group hanging off this end? That's right. And doesn't that mean at the other end, you're always going to have this little 3' hydroxyl hanging off this end, simply because of how you link them together? Think about it. Because I link them together the way I do, with the 5' phosphate always linking to the next nucleotide's 3' carbon, I'm always going to have a phosphate group hanging off this end and a hydroxyl group hanging out at the other end, right? Does that make sense? Think about that. So this is why DNA is considered directional. At one end, you have a phosphate group, and this is known as the 5' end, and that's known as the 5' phosphate. And at the other end, you're going to have a hydroxyl group hanging off. So this is known as the 3' end. Isn't that neat? And this is known as the 3' hydroxyl. And every strand of DNA and every strand of RNA has a 5' end with a phosphate group hanging off, called the 5' phosphate. And every strand has a 3' end with a hydroxyl group hanging off. That's called the 3' hydroxyl, right? And so this is exactly how DNA and RNA are made, from nucleotides. Nucleotides are the monomers, and a strand of DNA is the polymer. And by the way, the bonds that form between these nucleotides, these bonds that form when you link nucleotides together, are called phosphodiester bonds. We need to remember that these bonds are called phosphodiester bonds. And just to review, when you link monosaccharides together, those were called glycosidic bonds. When you linked amino acids together, those were called peptide bonds. When you linked lipids together, right? You don't link lipids together, right? And when you link nucleotides together, that's right, you form phosphodiester bonds. Now you get it. And by the way, look at this. This is called the backbone of the DNA. You see everything that's shaded? You see everything that's shaded in light blue or lilac or whatever color this is? Let me show you this. See everything that's shaded here? This is called the sugar phosphate backbone. Look, doesn't the backbone of DNA and RNA consist of a sugar phosphate group, sugar phosphate group, sugar phosphate group, sugar phosphate group? You see, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. This is called the sugar phosphate backbone. And what's poking out the sides? Those are the nitrogenous bases. Those are the nitrogenous bases, right? A's, G's, C's, T's, U's. You know, if it was a strand of DNA, it would use T's. If it was a strand of RNA, instead of T's, it would use U's, right? So hopefully that all made sense. Now, hopefully you fully understand what DNA looks like and why and how you string them together, to make a strand of DNA and a strand of RNA, and how, the components of a nucleotide. All right. So let's move on to exploring more about DNA and RNA. Now, most of the time, RNA exists as a single strand, just like this. However, what have you heard about DNA? Is DNA single-stranded or is DNA double-stranded? That's right, it's double-stranded, right? So you have two of these strands, right? Remember the strand which has a sugar phosphate backbone with a 5' phosphate and a 3' hydroxyl? Well, DNA usually has two of these strands, right? So you might be thinking of this, right? So you see on the left, this would be a double-stranded DNA, right? Two strands of DNA. And not only do they strand up like this, like they pair up, the two strands pair up, but they also form like this helix structure. You see, that's why it's called a double helix, because it's two strands of DNA and they kind of form this helical shape, right? Now, if you zoom in, I want you to zoom in into the, middle of this molecule here. You'll see that you have the blue part, the blue ribbon, which represents the sugar phosphate backbone, right? And what's holding the two DNA strands to one another? Look at this. The two DNA strands are held together by little tiny dots. What are these little tiny dots? What do they represent? They represent hydrogen bonds, right? So hydrogen bonds form between nitrogenous bases on the complementary strands of DNA. So if one strand has a T pairs with A on the complementary strand of DNA. G's pair with C's. A's pair with T's. Does that make sense? So you can see here, complementary pairing, complementary base pairing occurs between two strands of DNA. The A's always pair up with T's in DNA, and the G's always pair up with C's in DNA. This is known as complementary base pairing. However, in RNA, remember RNA is usually single-stranded, and in RNA, thymine is replaced by uracil or U. So in RNA, A's pair with U's instead of T's like DNA. A's pair with U's, but G's still pair with C's, okay? This is the big difference between how RNA and DNA work. And one more thing I want you to see here. Look at the two strands of DNA. You see the two strands of DNA? Notice how on one strand, this is the 5' end of one strand, and this is the 3' end on another strand. So what that should tell you is that the two strands of DNA, they go in opposite directions. So for example, if I have one strand of DNA, and here is its 5' end, and here's its 3' end, the complementary strand would always be going the opposite direction, right? With the 3' end here and the 5' end here, right? So this means that the two strands of DNA are always pointing in opposite directions. And the term for that is that the two strands are anti-parallel. So you should know that the two strands of DNA are anti-parallel. And here you can see where in the notes we're talking about the backbone of the two strands of DNA run in opposite directions. So the two strands are anti-parallel to one another. Now, you might be wondering, you might be saying, but Dr. D, I thought you said RNA is single-stranded, so how can it pair up? Why do A's pair with U's or G's pair with C's? Well, look, a lot of times, yes, RNA is single-stranded, but look what it can do. This is an example of a single-stranded RNA which folds onto itself and complementary base pairs to itself. Isn't that neat? This is an RNA called tRNA for transfer RNA. And notice how it's literally one strand of RNA. It's not double-stranded, but it looks double-stranded because it's folding back on itself. It's hydrogen bonding to itself. So G's pair with C's while A's pair with U's. Awesome. And here's our summary of key concepts for nucleic acids. I hope this was informative. Please let me know in the comment box below if you have any questions at all, if anything needs clarification. I appreciate you joining me, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr.